Okay. Going over DNA replication, we went over DNA transcription. When it comes to transcription, what you need to know is the location for transcription. Where does it take place? And the answer is simple in that sense. Any place where DNA is. So whatever DNA is located, that's where transcription will take place. So in eukaryotic cells, we have DNA in the nucleus. So the nucleus of eukaryotic cells. There is DNA in the mitochondria. So the mitochondria. And then in prokaryotic cells, there is DNA in the nucleoid of the cytoplasm. So nucleoid of the cytoplasm. The other thing you have to know, it's the products of transcription. So transcription is what makes up the messenger RNA, which is the copy of the recipe to make proteins. So the messenger RNA will be translated into a protein. Ribosomal RNA stays as ribosomal RNA. Transfer RNA stays as transfer RNA. Both these molecules are involved in translation, which is the process of protein synthesis. So these ribosomal RNA and transfer RNA will take the messenger RNA and translate it into a protein. But again, you do need to know the products of transcription and which of these products is translated. And the process of transcription itself is fairly simple. The enzyme involved in transcription, which is the RNA polymerase, is going to bind to the promoter region in the of the gene, and from there it will start to make a single-stranded copy of the gene. So the messenger RNA that's going to be made will be single-stranded messenger RNA. And then the process ends, and it will end by either a hairpin loop or a protein. We'll go into that again in just a second. Again, the anabolic enzyme, this is again an anabolic process because we're making something. Energy will be required. The energy is going to come from the nucleotides. In this case, the nucleotides will be the adenines, guanines, triphosphates, cytosine, and uracil, because this is RNA. The RNA polymerase does not need a primer, but at the same time does not have proofreading ability. So if it makes a mistake, the mistake stays. But that's okay because the mistake is just the copy of a gene. So if you're making over and over copies of the same recipe, and one of those copies does not have a right ingredient, it's okay because you have so many other recipes that are more likely okay, that are more likely fine. So it doesn't matter if there's a mistake. It's not that crucial. Like all other polymerases, RNA polymerase can only work in the 5' to 3' direction, which again is not that big a deal because we have two DNA templates. So one of them will serve as the template. So you should know what the promoter region is. In the case of prokaryotic transcription, the RNA polymerase has a little piece called the sigma factor that is going to help the RNA polymerase bind to the promoter. So once that happens, the sigma factor falls off, and the RNA polymerase can itself separate the DNA strands and start the copy. Now, because RNA polymerase has to work in the 5' to 3' direction, that means that it is copying the 3' to 5' DNA. So in this particular example, the DNA that's being transcribed is this top DNA right here, because that is the 3' to 5' DNA, which makes RNA polymerase go make the antiparallel strand 5' to 3'. If it was working on the lower one, it would have to be making a 3' to 5' messenger RNA, and it cannot do that. So the process of elongation keeps going. One of the points that is important to keep in mind is that this happens over and over again. It is many RNA polymerases transcribing a piece of a gene, which creates enough copies of the gene to be able to have many copies of the protein. A termination either happens via a hairpin loop or a protein. You should know what we mean by polycistronic messenger RNA, which is what we find in prokaryotes. It's a space-saving and time-saving strategy. Right there is the DNA, and this is the transcription with several pieces of that code for a polypeptide. Now, one thing to point out, I did say that the messenger RNA is translated into a protein. Technically speaking, that's not true. It's translated into a polypeptide. And then several polypeptides together will create a protein. However, it is okay to think one messenger RNA, one protein, as long as you understand that many times that's not true. Or where what the messenger RNA codes for is a polypeptide, which has to be bound to other polypeptides to actually create the protein. Okay. Initially, we did think one gene, one protein. Now we know it's a little bit more complex. It's one gene, one polypeptide, and several of these polypeptides will make the protein. But again, if it is it's simpler to think one gene, one protein, and most of the time it's not that big a deal. But, yeah. Yeah, go ahead, Joe.
What is it? These polypeptides, is this making like parts of like the cell wall? Like what, or is it just making genes?
Okay. All right. A polypeptide is making a protein. A polypeptide would be a short chain of amino acids. Keep in mind, a protein would have about 50 amino acids. So a polypeptide is going to have, let's say, 25, and it will have to join to another polypeptide to make the complete protein. So it depends on the protein. In some proteins, let's say, for example, in a protein, it will be several of the polypeptide ones that will need to join to make, let's say, protein 1. In other proteins, there are several messenger RNAs and several different polypeptides will have to make the final protein.
I thought this was like we're still talking about prokaryote. Like I thought that this was. I was thinking in terms of bacteria.
Oh yeah. No, this is bacteria also. Yes. Now, when you're thinking of, let's say, the 4 little amino acids for the cell wall of bacteria in the peptidoglycan, those 4 little amino acids would not be made in this manner.
Okay.
4 little amino acids is just not a long enough chain to really, have to be transcribed and translated in that manner. So now, having said that, the proteins that make the polypeptides, and let me rephrase, the enzymes that make the cell wall, those would be genes in the DNA of the cell.
Yeah. Okay. I was just confused about that. Thank you.
Yeah, no problem. The other thing we talked about was the post-transcriptional modification that takes place in eukaryotes, which does not take place in prokaryotes. And essentially what that meant was that the product of transcription was not messenger RNA, but a pre-messenger RNA that had to be modified in order to create the final messenger RNA product. And depending on how it was modified, that the final product could be a certain protein or another, or the recipe for one protein or another. So this gave the cell tremendous variability as to within one recipe, if you will, would create almost an infinite variety of proteins or tons of different proteins within just one gene. But at the same time, it's certainly given us a lot more complexity. So that's why bacteria are the organisms that are manipulated and not eukaryotic cells, because eukaryotic cells are a lot harder to manipulate. Okay, definitely make sure you can transcribe even a piece of DNA. And the question is, give the transcription of the DNA. The DNA is double-stranded, so the first thing you need to know is where is the promoter? And the question should tell you where the promoter is. So here it's telling you the direction of transcription is from left to right, which tells you the promoter is on this side, on the left-hand side of the DNA. And this is also to say, typically that's where we put the promoter because we read left to right, but a promoter could be on the other side. We're just more comfortable with this left to right reading of things because that's how we read. But a promoter could be on the opposite direction, in which case that would be the direction of transcription. Okay, and that happens a lot. Whereas you may have a promoter on either direction of a gene. So depending on where the promoter is, let me go back to this side. If the promoter is on this side, the DNA template that will be used to transcribe will be this one up here, the one above. That one will allow us to transcribe a 5' to 3' messenger RNA. However, if the promoter is on the opposite side, now the lower strand is the one that will be transcribed because that's the one that will allow us to make a 5' to 3' piece of DNA. And it will make a difference. The recipe will change. The sequence of nucleotides changes in one or the other. Keep in mind, if the one that is transcribed is the one on the top, if we transcribe this strand right here, 5', messenger RNA will read a U. Always remember that the complementary of the A in RNA is U, then G, U, C, C, G, C, 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 U. I'm going to stop there. I don't have room for the last two. So this will be the messenger RNA when the promoter is on the left and we're transcribing the 3' to 5' DNA strand. If we put the promoter on the right and we transcribe the lower strand, we end up with a completely different messenger RNA. Now we're going to have UU, the first two A's, then A, GGC, GGA, and then CAU. Again, I'm running out of room here, CAU, which is a completely different sequence from the one above. Okay, so it makes a huge difference. So pay attention to that. Any questions about that? So what we're going to do next is actually translate. So the last step in this process is translation, which means taking the messenger RNA and translating it, changing the language, in this case, a chemical language, from that of nucleotides to that of amino acids. So that's why it's called translation, because it's literally the changing of language, chemical language. All right, so the first question is, where does it happen? So we're back to the same question, eukaryotes versus prokaryotes. The answer in here is any place where ribosomes are located, because the location of translation is ribosomes. In the case of eukaryotes, we find ribosomes in the rough endoplasmic reticulum and in the cytoplasm. But don't forget that Eukaryotes also have ribosomes in the mitochondria. Eukaryotic ribosomes were 80S ribosomes. We saw that in chapter 3, whereas mitochondrial ribosomes are 70S ribosomes. Nonetheless, either 80S or 70S can translate. So this is where the location where translation takes place. Prokaryotes are easier. 70S ribosomes. Ribosomes are found in the cytoplasm and attached to the cell membrane. So know the locations of translation. In order to translate, cells have to take this nucleotide language, this sequence in the messenger RNA of A's and G's and C's and U's, and change that sequence into amino acids, which is what proteins are made of. So the question is, how does the cell know which amino acids go for each of these nucleotides? So this is the thought that scientists had when they were trying to figure this problem out. They knew there were 4 nucleotides. And so obviously 4 nucleotides, if you take them one at a time, it only gives you 4 letters in an alphabet that are supposed to code for 20 amino acids. Well, they're not enough letters. Okay, then if you start putting 2 amino acids together and you make the language or the code that gives you an amino acid, 2 letters, you still are not going to have enough. So you needed a language that had at least 3 letters together. If you combined all possible combinations of A, G, C, U on groups of 3, you end up with 64 combinations. So these are all the possibilities of arranging A, C's, G's, and U's in different manners. You have 64 possible different combinations. All you needed was 20, so this is more than enough to account for all 20 amino acids. So literally what they did is they created all these letters, they put them all together, and then they got together a whole bunch of scientists, and they assigned to each of the scientists a group of. I'm sorry. Are you talking to me, or is that just your microphone?
I'm sorry, that was echo.
Oh, okay. It's fine. Okay, so essentially they had to figure out what this group of 3 letters for which amino acid they coded. So they came back after however long it took to figure that out, and they put together what's called the genetic code. The genetic code is essentially a dictionary that tells us what the cell reads and translates into. So when the cell in a messenger RNA finds UUU together, it knows that tells the cell, here's where you have to put an amino acid called phenylalanine. UUC also means phenylalanine. UUA, UUG means leucine. And actually, leucine is also CUU, CUC, CUA, and CUG. So some of these amino acids have more than 1 letter that accounts for its translation. So that's a genetic code that we're going to use to translate. Notice that the genetic code accounts for, it's how come I told you, this is like riding a bike. If you've ever done this before, it'll come back. Notice that the language accounts for, this is the beginning and this is the ends. So there are 3 amino acids that code for stop, 1 that accounts for start. Okay, so this is all happening in the ribosomes. And ultimately, what the ribosomes are is they're just rooms, they're just places where this can happen. And places that can grab a hold of the messenger RNA and allowed for the transfer RNA to translate. The actual translator is the transfer RNA. That's the one that's really doing the translation part. What the ribosome does is just grabs a hold of the messenger RNA. So much so that in this picture, they're not even showing you the ribosome. that just, ribosomes, okay, so they're just there, just a place, interesting, that's basically all they do. It's a location. The actual translation is being done by the transfer RNA. The transfer RNA is this RNA molecule that it's described as a clover, like a four-leaf clover, but it has at one end an amino acid attached to it. At the opposite end, and this will be the opposite end right here, it has a 3 amino acid sequence that matches with the 3 amino acid sequence in the messenger RNA that in our genetic code codes for the amino acid. So let me repeat that. This is the messenger RNA in red. The messenger RNA has 3 amino acids that are AGU. AGU, if you look at the genetic code, AGU codes for an amino acid called serine. All right, so turns out the transfer RNA that has the complementary code for AGU, which will be UCA, will be the complementary sequence, has at the other end attached serine. That's the transfer RNA that carrying a serine. We called the messenger RNA, the amino acids, we call them a codon. The complementary will be the codon, which is in the tRNA. The tRNA carries the complementary sequence to the messenger RNA sequence that now we know translates for that specific amino acid. So the cell makes these transfer RNAs. These are made through transcription, and there are transfer RNAs that account for all of the amino acid sequence, anticodons. Go back to this one. So there will be a transfer RNA with an AAA sequence that can match to the UUU sequence, and that transfer RNA have at its other end a phenylalanine amino acid. There will be another one with an AAG sequence with, again, a phenylalanine attached to it, etc. So you can see how many different transfer RNAs are there. And the actual molecule that's doing the translation is the transfer RNA. Let's say this is another sequence. UAC is the codon. The anticodon will be AUG. UAC, UAC stands for an amino acid called tyrosine. So the tRNA that has the anticodon AUG sequence is carrying tyrosine. So this is how the whole process will take place. This is showing you the big and the small subunit of the ribosomes coming together. Do remember, review that the ADS of the ribosome is made of a 60S and a 40S subunit. That's the eukaryotic ribosome. Prokaryotic ribosomes are 70S with 30S and 50S subunits. So here you have the 50S subunit and the 30S subunit coming together to make the 70S subunit. And it is within those subunits that the messenger RNA is going to be inserted. Translation begins, it elongates, and then it terminates when it reaches a stop codon. So initially the messenger RNA binds to the small subunit. And notice that there is a start codon, which is AUG, which is where all genes start with an AUG. That tells the cell this is where the protein begins. So the AUG places itself into what we call the P site of the ribosome, and the next amino acid is on the A side of the ribosome. And here comes the tRNA with the anticodon for AUG, which will be UAC, and is going to bind with each other. Notice that this let's see, yeah, this is going to require energy. So we're going to spend energy in order, because this is, again, an anabolic process, in order to create this binding. Then comes the, let's see, okay. Well, that's all it is. Oh yeah, here comes the next, the other ribosome subunit put into place. Now the next tRNA that's going to show up, it's going to have the anticodon UUU. I think we said last time that one stood for serine, I believe. Okay, so the next thing that happens, now you have two tRNAs together, and we have one amino acid and the other amino acid close to each other. Then here comes an enzyme called peptidyl transferase. It's going to create a covalent bond between these two amino acids. So the enzyme, and it's in another PowerPoint, peptidyl transferase. Next thing that's going to happen is that the ribosome is going to move both subunits to the next portion, and this tRNA is going to leave. Okay, so we have the peptide bond formed by peptidyl transferase. The ribosome moves, and notice that what moves is the ribosome, not the messenger RNA. And the tRNA for the first codon will leave. The next tRNA comes into place, and now we have the first two amino acids together. The ribosome moves to the next codon. Another peptide bond is created, and the whole process continues in that manner. So that here it is, the first tRNA is leaving, ribosome moves, and now the next nucleotide is ready to receive its tRNA. Here's the next tRNA. The two amino acids come together. Now we have three amino acids put together in a chain. The whole ribosome moves. The first amino acid leaves, and now we're ready for the next amino acid to come in. And so the cell keeps doing these, creating longer and longer chains of amino acids until we hit the stop codons. The stop codons have no tRNAs. So at that point, nothing will happen, and the ribosome begins to disassemble, and the tRNAs and messenger RNAs disassemble, leaving out just the polypeptide, the chain of amino acids. Now, what do you need to know from these? You don't need to know all of the whole process. You do need to know the location where this happens. You need to know the subunits of the ribosomes. You need to know the enzyme that makes the peptide bonds, the peptidyl transferase. Given a messenger RNA, you should be able to translate it when given a genetic code. So you will definitely need a genetic code to translate. And we'll do that here in just a second. Another thing to keep in mind is, again, the cell doesn't need one copy of the gene, of the protein, of the polypeptide. It needs many, many copies. So there will be many ribosomes sitting on a messenger RNA, and all of them moving and translating that polypeptide, so that at the end of the day, there's many copies of the polypeptide being made. Again, termination happens when the stop codons are arrived. So when it comes to translation, back to this right here. Given a double-stranded piece of DNA, you should be able to transcribe and translate the DNA. So, and again, in this case, doesn't have the promoter, but we're going to assume the promoter is on the left. This is telling you that this is the 3' end, which means this is the 5' end, 3', 5'. So when it comes to transcribing, we're going to transcribe the 3' to 5', DNA strand so that we can transcribe it 5' to 3', and the messenger RNA will read AUG, GGG, CGC, AUC, and UAA. So that's the messenger RNA. You should know the enzyme that can transcribe, and the location of transcription. Then you want to be able to translate. Okay, so AUG. Again, you're going to need a genetic code. I don't want to go back to the slide because I'm going to lose what I just transcribed. If you look at the genetic code, oh, well, too late. Okay, let's see. AUG was the first start. The start codon. It actually codes for an amino acid called methionine. I can have this here. Okay, there we go. All right, so it was AUG, and that will be translated into methionine. Then CCC, which was GGG, and GGG is, just a second, there we go, glycine. So glycine will be the next amino acid in that sequence. GCG was the next one, so GCG will be translated into alanine. So we had methionine, glycine, alanine. TAG. TAG would be AUC. So AUC right there, isoleucine. And then finally UAA, and UAA is a stop codon, so we have a stop. So translating that messenger RNA will give us methionine, glycine, alanine, and isoleucine as the chain of polypeptides. You should be able to do that. You should be able to, given an amino acid, given the genetic code and a messenger RNA, you should be able to translate. Okay. Questions about translation? Again, this is showing you translation in eukaryotic cells. Yes, that chart will be on the test. So if there is a question that needs a genetic code will be together with the question. So there will be a picture of the code with the question. Any question that needs a genetic code will have the picture. Yeah, and the genetic code will be the same as the one that is here in the PowerPoints, which is the same as the one in the book. So it's so you're familiar with that one, because there's a few different ways to put it together, and so anyway. Okay. Let's see. Now this picture is showing you location of translation in eukaryotes versus prokaryotes. One thing to note, this is very good because it's showing you the messenger RNA processing, transcriptional modification, messenger RNA. Messenger RNA has to be transported into the cytoplasm where the ribosomes are going to make the polypeptides. In the case of prokaryotes, you can see why prokaryotes will grow so quickly and be metabolically so active so fast is because transcription and translation are happening at the same time. The cell doesn't have to wait for transcription to begin translation. You can see here how this messenger RNA is still being transcribed, and before the transcription is even over, the ribosomes already begin to sit on it and begin to translate it. So the whole process goes very, very quickly because of that, because transcription and translation can go on at the same time. Oftentimes, ribosomes are the targets for antibiotics. That's not unusual. The problem with making ribosomes target of antibiotics, which we do all the time, there are many antibiotics that target ribosomes, is that yes, ribosomes in prokaryotes are 30S, 50S, 70S ribosomes. In eukaryotes are 40 and 60S. However, remember that eukaryotic cells have mitochondrial ribosomes that are the same as prokaryotic ribosomes. So any drug that targets prokaryotic ribosomes will damage or could damage mitochondrial ribosomes. So those antibiotics will be a little bit more toxic. Also, what those antibiotics do is typically slow down the cell. It doesn't really kill it. So they don't really stop protein synthesis completely. It just slows it down. Okay. Now, the last thing that this chapter looks at is control of gene expression. And essentially, what it explains how cells control which genes are being transcribed and translated, which is what gene expression is. So when we talk about genes being expressed, we're talking about genes being transcribed and translated. So what this is saying is, at least in prokaryotic cells, most of the genes are being expressed at all times, which makes sense because prokaryotes have very small genomes. So they don't have much room for useless genes. However, when it comes time to regulating, saying we're not going to express this gene, that regulation occurs at the transcription level, not at the translation level. In other words, what's going to be stopped is going to be transcription, not translation, which makes sense because why transcribe something that is not going to be translated? Why waste time and energy making the copy of a recipe that is not going to be cooked? So instead of stopping the cooking process, what is stopped is the copying of the recipe process, which makes a lot more sense. So most of the regulation occurs at the level of transcription, which means that what's going to be regulated or stopped is an RNA polymerase. So we have constitutive genes or enzymes, which are the ones that are transcribed and translated at all times. These are the types of enzymes the cell needs all the time. Some enzymes can be inducible, meaning that they're only going to be expressed when the cell needs them. Otherwise, they're always off. So they are off until they need it, and then they're turned on. Repressible enzymes are enzymes that are exactly the opposite, always on until they're not needed, and then they're turned off. They can be repressed, versus the ones that are off can be induced or turned on. So they're inducible enzymes and repressible enzymes, and that's the difference between. And in order to understand gene expression, we have to understand how genes are organized in a that the same as inhibited. Yes, repression means inhibition. Correct. Yes, the repressor is something that inhibits. Exactly. Yes. So this is an enzyme that can be inhibited, or its expression can be inhibited. The cell can stop making it. Whereas the other one is not being made, but it could be turned on and made if needed. So the way genes are organized in a eukaryotic cell is we have a promoter, and then we have the genes. And remember, we said that instead of having a promoter for each gene, if we have a group of genes that had something to do with each other, the cell will have clustered them together and put them under one promoter so that they're always transcribed together. The operator region is the control region right here. This will be the on-off button, if you will, which is strategically located between the place where the RNA polymerase is going to sit and the coding part of the gene is located, so that the operator region can say, yeah, go ahead and go on and transcribe, or no, stop, you cannot go on and transcribe. So the operator is the control region. The entire thing is the operon. This regulatory gene right here refers to the gene that makes a protein that is going to sit on the operator and turn the gene on or off, depending on the gene. So that's the regulatory gene for the regulatory protein, which is going to sit on the operator. Okay, so we have an example of an inducible operon. Those are the ones that are off. We can turn them on if we need them. The example is that of the lac operon, and what's going to turn it on is going to be an inducer. Repressible operons are the ones that are always on, but could be turned off if we don't need them, so that if we don't need them, we're going to need to activate a repressor. The tryptophan operon is a repressible operon. So we're actually going to talk about the tryptophan operon first. So the tryptophan operon refers to all of the enzymes involved in the synthesis of an amino acid called tryptophan. So each of the 20 amino acids requires a different biochemical pathway to be synthesized with different enzymes. In the case of tryptophan, this is a very common amino acid in the proteins of bacteria, so they're always making tryptophan, always needing tryptophan. So this is a gene that is always on, but if for whatever reason it has to be turned off, it could be turned off, it would be repressed. Turns out there is would inducible, repressible be considered feedback systems? No, because, give me a second. No, because a feedback system would be, you know what, it is. Yeah, it is. Yeah, it is a feedback system. I was thinking about, yes, the end product is ultimately the one that's going to come back and turn it on or off, which is what a feedback mechanism is. A feedback mechanism is one in which the end result, the end product, is what controls whether the system is going to go on or is going to be stopped. When there is too much of the end product, then the end product itself is going to turn off the whole system. If there are not enough of the product, then the whole system keeps going and keeps making the product. Okay, so yes, it is a feedback system. Absolutely, it is. So the end product is going to be tryptophan. That's the end product of this whole process. So normally, the cell is in a continuous need of tryptophan. So it's making tryptophan and immediately uses it in the proteins that are making. So there's never enough tryptophan around. Because there's never enough of it around, the cell keeps making more. Okay, there is a repressor protein that is laying around, but it's inactive. So it's always out there, but it's not working because it's in an inactive form. What activates that inactive repressor is tryptophan. But tryptophan, it's never around because it's always being used. So the minute it's made, it's being used, so it's being removed. So it's never hanging out. If for whatever reason, whether it's because the cell slows down and it's not making as many proteins, if for whatever reason, tryptophan begins to accumulate, the cell is not using it anymore. Now it begins to hang around, and it can bind to the repressor and activate it. And now the repressor can sit on the operator and physically blocks the RNA polymerase from transcribing. So it's essentially a physical block. Okay? So that's how the gene will be turned off if the products of the gene are not needed anymore. The end product accumulates, end product binds to a repressor and activates it. The repressor can now sit on the operator and block RNA polymerase. So that's the repressible gene. The inducible gene is on the lac operon. And in this case, we're going to need an inducer to turn the gene on. The lac operon was the first operon that was figured out. And it's a very useful operon. This is how it works. Lac operon is short for lactose operon. This is the genes that are involved in the catabolism of lactose. So that if the cell wants to catabolize lactose, first thing it has to do is bring lactose into the cell. So transport. It's going to need some type of protein that transports lactose into the cell. And lactose is a disaccharide. It's made of galactose and glucose. So galactose and glucose bonded. So now that it has to break them apart and separate the galactose and the glucose. That is another gene that needs to do that. Glucose, cells can make energy from glucose without any problem. That goes into glycolysis and everything else we looked at in chapter 6. So glucose is immediately shuttled into glycolysis. Galactose, on the other hand, does not fit into glycolysis, so it will have to be changed into glucose. So now we need another enzyme to change galactose into glucose, and now it can go into glycolysis. So we need a transport enzyme. We need another enzyme that breaks up galactose and glucose, and then a third enzyme that converts galactose into glucose so that it can be catabolized and used for energy. So those are the genes that are part of the catabolism of lactose. If you have lactose around, if you're going to use lactose, you're going to need all three genes. So that's why the operon controls all three genes together. This is the deal, though. Cells prefer glucose. In the presence of glucose, the lac operon is turned off. So since glucose is usually the source of energy, usually the lac operon is turned off. And it is turned off by a little repressor protein that sits on the operator and physically blocks RNA polymerase from moving on. Okay. What makes that gene be turned on? Now, none of these systems are completely on or off. Even if we say this system is off, in reality is mostly off. Once in a while, the block is not complete because this repressor binds and comes off, binds and comes off, binds and comes off. So it's not permanently attached there. So one of those times when it comes off for a few milliseconds, RNA polymerase can all of a sudden real quick go on and transcribe. So if all of a sudden there is lactose present in the cytoplasm because the transport enzyme found lactose outside and brought it in, okay? Lactose itself is what's going to bind to the repressor and inactivate it. Now the repressor cannot sit on the operator. Now the promoter and RNA polymerase are free to start transcription. Okay. So again, this is a feedback mechanism in that the what the gene is going to be acting on is what's going to activate the gene. So in the presence of lactose, lactose is the inducer. It's going to bind to the repressor and allow for transcription. The next PowerPoint attempts to explain why is it that in the presence of glucose, the lac operon doesn't work. So as long as the cell has glucose, and glucose is the preferred source of energy. So as long as glucose is there, even if there is lactose present, the cell is not going to touch the lactose. It's going to go for the glucose first. And that has to do with something called catabolite repression. So the presence of glucose essentially does not, through a series of events, does not allow the RNA polymerase from binding in the promoter. So if glucose is present, RNA polymerase will not bind to the lac operon because the glycolysis is removing a little molecule that's needed for RNA polymerase to bind to the promoter. So as long as glucose is present, RNA polymerase will not bind to the promoter of lac operon. So even in the presence of lactose, if glucose is also present, RNA polymerase will not be able to work. So that's what catabolite repression means. It means we have a sugar that is repressing the transcription of a gene. Yes, CAMP is yes, it is a second messenger. So this is essentially what's happening. A protein called CAP binds to the activator site, and it's needed for RNA polymerase to bind to the promoter. However, in order for CAP to be made, CAMP must be present. So CAMP makes CAP possible. Cyclic AMP is the precursor to ADP, which is the precursor to ATP. So we go AMP, ADP, ATP. If glycolysis is going on because glucose is present, ADP, instead of going to make to the AMP that's going to be turned, sorry, let me put it this way. The AMP will be used to make ADP, which will be shuttled into glycolysis. So let me put here glycolysis. There we go. So glycolysis will be using the ADP to make ATP. So this cyclic AMP is going to go in this direction instead of going in the direction of making the CAP protein. And that's why cyclic AMP is not available in the presence of glucose to make the CAP. That's essentially what's happening. Cyclic AMP is being shuttled towards glycolysis to be made into ADP and then used for glycolysis. So will not be available for the CAP protein. So that's essentially why, as long as glucose is present and glycolysis is going on, the cell is not going to touch lactose. So what that means is that if we have glucose, okay, so let's look through all the scenarios. If glucose and lactose are absent, then everything is off. There is the, yes, you have the AMP, and yes, you can have the CAP, but the lactose is not present to get rid of the repressor. So what happens in this instance, so these are the genes, is that RNA polymerase can bind to the promoter. However, the repressor is sitting there, not allowing it to go on. So there is no transcription. Glucose not present and lactose present. Okay, this will be the only scenario in which the lac operon is going to be transcribed. If glucose is not present, now we have the CAP-CAMP available for RNA polymerase to bind. So RNA polymerase can bind. Lactose is present to remove the repressor. So now the little repressor right here is being blocked by lactose. And so there's nothing to prevent RNA polymerase from transcribing. So this is the only scenario that gives us transcription of the lac operon. Glucose present and lactose not present. Okay, so there's no lactose and there is glucose, which means the CAMP cap cannot happen. Which now what that means is that the repressor is here. And on top of that, RNA polymerase cannot bind. Okay, so even if it could bind, the repressor will be in its there stopping transcription. Glucose and lactose are present. Okay, now we have both. So we don't have the CAD-CAMP, but we do have the repressor. This can be. And again, you don't have to know, I mean, on this much detail. I just thought that it is if you want to understand it, it is easy to understand if you explain every single scenario. Let's see, so glucose is present, so RNA polymerase cannot bind, and the repressor is removed because of the presence of lac. It doesn't matter. Oh, good. I'm glad you understand it. Yeah, this is a very clever system when you think about it. So now the repressor is removed because lactose is present. However, RNA polymerase cannot bind because the cyclic AMP is not there because glucose is present. So anyway, transcription cannot take place. Okay, now part of the reason why I went into all these is because the lac operon ends up being a on-off switch that we get from bacteria. And it is very useful because we can turn it on by removing glucose and putting lactose, and we can turn it off by removing lactose and just giving the cell glucose. So it's a very easy way to turn genes on or off by using the lac operon. It has become a switch, an on-off switch. We cut the lac operon, we add it to a gene, and now we put that gene inside a cell, and we can turn that gene on and off by just the levels of lactose and glucose. So that's why it's become a tool. It's become a tool, a little tool that is used to turn genes on and off, control genes. So that's part of the importance of the lac operon. The other importance is that what scientists said was, okay, if this is the way that prokaryotes control their genes, this is probably the way eukaryotes control their genes also. And of course, they're right. Turns out we control our genes in similar manners also. So it gave us a model. Yeah, exactly. That is exactly it. Yes, this is exactly a model that opened the door to understanding how human beings control their genes. And this opened the door to epigenetics, because for the first time we realized that something that comes from the outside, something we eat, could control gene expression. And that's what we call epigenetics, the environment controlling our genes. And it is a huge field nowadays. Okay. So just know what we mean by that gene expression is being regulated at transcription level, know what constitutive genes are, what's an inducible operon versus a repressible operon. Remember the two examples we have for each. The inducer in the lac operon is lactose, the repressor on the operon is tryptophan. And then given any of these scenarios, you should know what's happening. So here you have a no messenger RNA being made. You know that lactose is absent, and there is no messenger RNA being made, that explains lactose being absent. Cyclic AMP is low, which means glucose is present. So glucose is present and lactose is absent. That's why the lac operon is not working. Here you have both are present. No messenger RNA synthesis, so no says it's slow. You can just put no there. So that means that this will be low also because glucose is present. All of a sudden, cyclic AMP is high. That makes sense because glucose is absent. There is no messenger RNA, which means that lactose is absent also. And then here's the only scenario in which you have messenger RNA is when glucose is absent, lactose is present. Okay. All right. Any questions? I'm going to go ahead. I know we kind of gone through the hour, but I'm going to go ahead and let's push it to 10 o'clock and let's go to chapter 8, which is not a bad chapter. For one thing, let's see. Yeah, it's only 64 slides. The other one was 108. So you can see this is a lot shorter. All right, chapter 8. This goes into bacterial genetics. Essentially, what this chapter answers is the reasons for bacterial diversity. Because in theory, there shouldn't be bacterial diversity. If bacteria clone each other, then we should have no strains. We know that's not true. We know that bacteria are strains within a species. So that means there are ways in which bacteria are going to change, slightly change their genetics. So the question is, how do they do that? So this diversity arises from changes in DNA. There are two mechanisms that are going to change DNA, vertical gene transfers or horizontal gene transfers. Verticals are easy. These are mutations. Vertical gene transfers are the ones that are made and transfer from mother cell to daughter cell. Horizontal, that involves three mechanisms that we're going to talk about. So vertical gene transfers is transfer of modified genetic material from mother to cell. Horizontal is when two bacteria that are completely unrelated to each other transfer genetic material to each other. Exactly, Sexpila is one of them. Exactly, you got it. Yep, that's one of the mechanisms. It's called conjugation. Yes, that's one of them. That's supposed to be the DNA that's mutated, the cell divides, and now mutations are passed on to daughter cells. That will be vertical gene transfer. This is organisms A and B. There is an exchange of small pieces of DNA from one to the other, and so that will be a horizontal gene transfer. Okay, so genotype is the genetic material of an organism. Phenotype is the expressed genes, the ones that are being made, transcribed and translated. All right. So mutations and horizontal gene transfers can change the genotype of an organism, and that may, not always will, but it may lead to a change in phenotype. So the organism may acquire the ability to make new proteins through these gene transfers. Okay, so it may or may not happen. All right, so you should know the two mechanisms that can cause diversity in bacteria: vertical mutations. Horizontals are actually conjugation, transduction, and transformation. Okay, so we're going to talk about mutations first. So actually, let me go ahead and go over there. And all right, so may not have to do with duplication of gene codes. They may not have, in the case of mutations, it has to do with the mutation not changing anything. And yes, it has to do with the duplication of gene codes. Exactly. You got it. It's a silent mutation. Exactly. It's silent mutation in which there is a change, but the change does not change the amino acid. Absolutely. Yes, that's exactly why. Yeah. So that's why it may change, but it may not. Correct. So diversity has to do with vertical gene transfer, and that would be mutations. I might as well put all the possibilities here. The other one is called horizontal. And this one has three mechanisms: conjugation, transformation, and transduction. So one thing I wanted to point out, the next test has a lot of trans words. And every time you get to a T word, slow down, make sure you understand which word they're talking about, because we're going to deal with transformation, transduction, transcription, translation. So 4 words that sound very, very similar. So slow down and make sure you understand what is being asked. So I wanted to point that out. Okay, so mutations, it's changes in nucleotide-based sequence of a genome. Okay, almost always they're bad. Deleterious means deadly. So almost always they're going to kill the organism. Rarely a mutation would lead to something new that improves the organism. That's the first thing to remember. There are 2 types of mutations. Spontaneous, based on the cause, what caused the mutation. Mutation may be spontaneous, which is a mistake in DNA replication. Remember, DNA polymerase does not make mistakes. It has proofreading ability. So I should say it makes very few mistakes. So 1 in a billion nucleotides, there may be a mistake in there. Okay. So spontaneous mutations are rare because it involves a mistake in DNA replication, and DNA polymerase very seldom makes mistakes. Induced mutations, on the other hand, the cause of the induced mutations are mutagens. And mutagens are agents that increase the rate of mutations, so make mutations more possible. It actually increases the rate to 1 in 1,000 nucleotides, 1 in a million, depending on the mutagen. Some mutagens are so bad that create lots of mutations. Others are not quite as bad, but still are worse than DNA polymerase. Okay, so we're going to look at spontaneous mutations first. And a spontaneous mutation is going to change the DNA. And how do we know it has changed? What are we comparing it to? In the case of bacteria, we compare it to wild type. A wild type means this is the most common strain of an organism that has been identified. So, for example, E. coli. When we began collecting E. coli and identifying it, the most common strain of E. coli that would have appear over and over and over again, that's what we would consider the wild type. Anything that was a variation from that most common E. coli was considered a strain. So having said that, the concept can get a little tricky. But in general, we do nowadays have pretty much the genetic codes of most of the common organisms, so that it is easy to know, okay, this is that strain, this is this strain, this wild type, meaning common strains. Okay, so any variation from the wild type is a mutation. A spontaneous mutation is if that variation was due to a mistake in DNA replication by DNA polymerase. Now, a reversion, we're going to talk about a reversion at the end of the chapter. A reversion is a second mutation that reverses the effect of a first mutation. So The first mutation created a non-working protein. Okay. A second mutation somehow turns that non-working protein into a working protein. What a second mutation never does is it fixes the first mutation. That first mutation remains, but a second mutation somehow, and we'll talk about a couple of possibilities that would make that protein work again. Okay, so that's a reversion. Knockout mutations are mutations that give you a non-working protein. And this is what typically happens. So most mutations are going to be knockouts. Point mutations is a mutation that involves reversions on reverse anything. No, they don't. Well, they reverse the result. They reverse the result of the first mutation, but they do not reverse the first mutation. So let me just give you an example. One thing that a second mutation could do is, for example, change the proteins that fold the mutated protein, so that the proteins that are going to help to fold the mutated protein now help it to fold properly. If that helps in any way. Another thing could be, let's say the first mutation created, I don't know, let's say created an area that all of a sudden is hydrophobic. It used to be hydrophilic, so it has to be hidden and twisted. The second mutation may create an area that is hydrophilic in another part, so that now the protein folds in such a way that it could potentially work better. So let's say now it's closer to the initial folding. If hopefully that makes some sense right there. So that would be the result. Protein folding, beta pleats, yes, absolutely. Alpha helices, beta pleats, that's the second, correct. That's what we're talking about. Not only that, but also the folding on the third conformation. So the primary folding is just the linear, secondary folding, alpha and beta sheets, and then on top of that, it could have a three-dimensional folding. So it could also affect that. So yes, it is affecting all that, so that it refolds it in a way that it could work a little bit, if not completely sometimes. Okay, let me say these. Reversions are incredibly rare, extremely rare, in the order of maybe 1 in a trillion or more. Okay, so it is an extremely rare event. It is possible, but it's extremely rare. Point mutations involve changing one nucleotide. Most spontaneous mutations are point mutations. Base substitution is a point mutation in which one base is substituted by another. So a point mutation could be substituting one base for another, so substituting an A for a G, for example. A point mutation would also be adding a base or deleting a base. And it's a point mutation because you're dealing with you're changing. It involves one nucleotide. So you add one nucleotide, you delete a nucleotide. That's also a point mutation. Okay, so there are 3 types of base substitutions, which are again point mutations. And notice that here we're talking about spontaneous mutations. These are the kinds of mistakes that DNA polymerase can make. It can substitute one base for another, but that substitution changes nothing. So that even though the base has changed, the amino acid has not changed. That's a silent mutation. A missense mutation is when the base has changed, but that does change amino acid. A nonsense mutation is when the base changes and that change creates a stop codon. These kinds of mutations, the nonsense mutations, typically are knockout mutations because it's creating a shorter protein, one that has a stop codon earlier than it should have had. Missense mutation, the protein may work. In a nonsense mutation, it never works. All right, so these are examples. Same for all cells. Yes, these are the same for all cells, eukaryotes and prokaryotes. The difference is when we're dealing with prokaryotes, because of sheer numbers, these rare mutations are possible. When you have 300 million cells per mil in a culture of bacteria, and you have 3 mils of culture, these kinds of mutations are possible just from sheer numbers. There's so many of them. In a eukaryote, extremely rare. In a prokaryote, it's not rare at all. So this is the wild type. This is the normal, if you will, nucleotide in this particular protein. This is what it should give us. It should give us, so this is 5 to 3, this is 3 to 5. So this is the coding, one that's going to be used to transcribe. So ACA will give us UGU as the codon. UGU codes for a cysteine. That's the wild type. Okay, so there's a mutation that changes that last C for a G, which means that now the codon will change from a UGU to a UGC right there. But UGC also codes for cysteine. So nothing has changed. But that's fine. This is a change in genotype, which gives us no change in phenotype. Next one, the A changes to a C, which now changes U to a G. And instead of a U, now we have a G. So before we had a UGU that coded for cysteine, now we have a UGG that codes for tryptophan. So if we change a cysteine to a tryptophan, that's a missense mutation. This protein may or may not work. It depends on how different these two molecules are. Last one changes, again, an A for a T. So we're going from a UGU to a UGA. A UGA means stop. So a protein that should have had more nucleotides, so more amino acids after that, all of a sudden is cut short. Nonsense mutation. This is knockout. This is not going to work. Okay. So that's one example. Here's another example, same thing. This is the nucleotide that is being changed right there. So in this case, it will be phenylalanine that may not or may not be changed. So you can go through these and see how actually this case now is the next one that is being changed. So these are the examples, and another set of examples. However, this slide also gives you the frame shift insertions and deletions. So in this case, a frame shift insertion is when there is an extra nucleotide inserted, and notice what that does. From the point of the insertion, every single nucleotide changes. So from a phenylalanine, tyrosine, alanine, and arginine, now we have phenylalanine, isoleucine, cysteine, threonine. Everything changes. This is a knockout. There's no way in the world this is going to work. This is the opposite, removal of a nucleotide, which means the entire reading frame changes. And again, every single nucleotide after the deletion is going to be changed. This is another knockout. There's no way in the world this protein is going to work. So knockout mutations, these three are knockout mutations. The knockout. Obviously silent, this one is okay, nothing happens. And missense may or may not work. So out of the 5 different possibilities, 3 of them will give you a non-working protein. One will give you a working protein, the other one may give you a working protein. So this is why we say mutations seldom give you something good and novel. Most of the time, they kill this. Okay. All right, so that's just, again, deletions and insertions. Okay, let me just go through transposomes and we'll probably stop there. Transposomes are little pieces of DNA that can jump from one place to another. They're also called jumping genes. And they move from one position to another. And when they move, you can see here, this guy moved from here to here. They disrupt genes. They turn off genes. They can also turn them on. So these are transposable elements or jumping genes. They're just little pieces of DNA that have the ability to jump from one piece of DNA to another. They have not been found in humans. They have been found in plants, especially in corn and in bacteria. Okay. All right, so name the type of mutation. It's just a matter of time. Yeah, you may be right. It may just be a matter of time before we find transposons in humans. Yeah, maybe. Right, maybe. Yes. Yeah, they have not been found yet. I don't know, we'll see. But they're obviously pretty bad. You should be able to figure out the kind of mutation, given an example of a mutation. So here we have the wild type, and the first A is changing to a C. So that's going to change a UUA into a GUA. And it's just a matter of now going to the genetic code to see what the change is and to determine what kind of mutation that is. Okay. All right. So let's stop here and we'll meet again on Monday. Okay, let me look at when is the when is the next test? That's what I was gonna. Can you post lab 7? Yes, let me post lab 7. I can post.
Isn't next week? It's the week after.
Oh, really? Oh, perfect. Really?
So
perfect. We have time.
Yeah.
Okay, so Monday we'll go over lab 7, and then Thursday we'll wrap up chapter 8.
Yeah.
Yeah, that's perfect. Okay, that's very, very good actually. All right, so let's do that. We're gonna have a little break. Okay, good deal. So yeah, I'll post chapter 7 and I'll post this recording also. Very good.
Is there going to be a discussion board question for this module, for this lecture module?
Yes, absolutely, absolutely. Yes. Yeah, I'm gonna go ahead and finish out those discussion questions. Yeah, let me do that. Discussion questions. Yeah, absolutely. Yeah, I'll do that. Okay. Yes. Yep. See you Monday. Okay. Have a good weekend. You too. Bye bye.